Pestalodiopsis microspora is a kind of wonder fungus. As a member of the phylum Ascomycota, it is capable of reproducing sexually. However, typically it reproduces asexually via its conidia. It can be found in the tropical and subtropical regions all over the world. Many isolates have come from just beneath the bark of various yew tree species. However, it has also been isolated from the bald cypress tree, which can be found locally here in Houston. Other strains have come from the leaf litter of rainforests in South America. While it is not harmful to many of the trees in which it is found, it is pathogenic to some ornamental and fruiting species. Loquats, Japanese headcoat, Chinese flowering pinks, kiwis, and pecans are all susceptible, and disease caused by P. microspora is being discovered on new species. Besides its pathogenicity, P. microspora has been discovered to be helpful too. Scientists have been studying the ability of fungi to break down plastics since the 1960s. In 2012, a group of students with Yale University, under the leadership of Dr. Gary Strobel, obtained several strains of Pestalodiopsis microspora from leaf litter in the Yasuni rainforest in Ecuador. While all strains were able to grow using plastic as their only food source, several of the strains did so remarkably quickly and without oxygen. The speed at which this was done, and the fact that it was done in the absence of oxygen, opens the door to this technology being used in landfills, where much of the plastic is under so much refuse that oxygen is not available. Although since the Yale study that publicized this finding in 2012, no suggestions have yet been made for its practical application in real-world settings. P. microspora can also be used to make a very important cancer drug, more efficiently than current methods use. Taxol is one of the two most commonly used cancer drugs, and the current process by which it is made from a tree of the Taxus genus involves the time-intensive growing of the tree and subsequent harvesting of the bark, which ultimately kills the tree. Additionally, each tree yields only a small amount of Taxol. Fungus grows much more quickly than trees, and the yield is higher with approximately 3 weeks' time. The implications of these improvements in production time could potentially reduce the cost of treatment for patients affected by various carcinomas and patients suffering from Kaposi sarcoma as a complication of the AIDS virus. Additionally, more efficient production would enable more widespread access to treatment with Taxol for those afflicted. P. microspora is also able to incorporate genes from the species it is inhabiting, giving it tremendous genetic variability through horizontal gene transfer. As it is posited that this quality is what allowed it to inherit its ability to produce Taxol from the yew. But an ability to adaptively mutate means that the fungus could potentially find new places to live if introduced to a new environment, causing environmental havoc, either by escaping the lab or being introduced into a landfill. Students are being encouraged to work with Pestalodiopsis microspora, especially in regards to its potential application as a plastic eater. However, it is important that we proceed with caution and recognize that while this wonder fungus could help us solve a couple of major issues with which we are faced, there is no magic cure, and we must all work to prevent the causes of these problems in the first place.